Hello everyone, my name is Jan Fulsek and I would like to welcome you to my poster on resting state dynamics from the perspective of structured flows on manifolds. The resting state is an uncontrolled but highly structured paradigm which has interesting spatial temporal patterns relevant both functionally or as biomarkers across various brain states and disease. What we would like to do is put forward these structured flows of manifolds as prime object of interest, which helps us to bind together the theory and the modeling and the empirical data features, in this case the dynamical ones such as coactivation patterns, dynamic functional coactivity, and so on. Let us start with the network model. We employ whole brain network modeling, which is based on connectivity derived from DTI data based on some parcellation, and where each of the nodes is equipped with dynamics, in this case governed by a bistable Monbiot-Pazuroxin model. This model has two stable states, upstate stable focus and downstate stable node. That means that on the fast time scale can exhibit oscillations in the upstate and noise-driven switching between the up and down states. On top of that, we process this simulated data with a bold observer model, which translates the fast time scale firing rate here for each of the nodes to a bold signal as comes out of fMRI. So let us first study how the connectivity structure shapes the dynamics, in this case by breaking the symmetry of the connections. When we scale the global coupling parameter from 0, which corresponds to the disconnected model, to larger values where the input of the network node is saturated by its neighbors. In the middle of these symmetrical cases, we arrive at a working point or working regime where the model exhibits dynamics characteristic for the resting state. So that's this fluid changes between states with similar functional connectivity. Interestingly enough, in the same part, the dynamics of the model as measured by variance explained by the first few components of the PCA of the trajectories in the state space becomes also low. As I said, the dynamics in this working point regime is similar to what we find in the empirical data here, and it starts exhibiting these characteristic cascades on a fast time scale, which also corresponds to the coactivation events as measured on the bold signals. And we can find exactly the same thing with these cascades and the bold coactivations reflecting the functional connectivity fluctuations, also in the simultaneous EEG and fMRI data. Now, let us look or reinterpret the similarity across time and the dynamic functional connectivity as visiting different subspaces of the system. So in this case, we start from the edge time series, which corresponds to the functional connectivity at different time frames, and we cluster this together and look at each of the clusters and to what subspaces these correspond in the state space of the actual system. Right? So we go from the bold to the model state space. And what we have found is that these clusters, so for example, those which correspond to the coactivations, they concentrate in a certain parts of the subspace, whereas the intermittent periods, which are dissimilar in terms of functional connectivity, correspond to different parts of different parts of the state space. This figure basically recapitulates the same thing, as in we have this part of the manifold, the dark blue one, which is the low activity, right, where most of the nodes are in the down state, whereas here is the high activity subspace, which also exhibits, in this case, the coactivations, and on this plot, the cascades. And what the model does slowly translates from each of these subspaces, and each of those is associated with this specific flow. We have also looked at the mechanistic structure which is behind this behavior, this flow, and we have systematically analyzed the fixed point skeleton on which these trajectories evolve, so that we have here, right, these colorful dots are the particular fixed points around which the system travels. And also we look at all the available fixed points around which the system trajectories can evolve, and how these change with the coupling strength. These the fixed points, those which are for example lost here as we increase the G, which you can see from this figure, these depend on not only the position of the node in the network, that is how well it is connected, but also to the state of its neighbors. Why am I saying this? Because this is the same principle is behind changes or shifts in the characteristic frequency of each of these nodes in the upstate. So if again we plot this characteristic frequency against int strength, we get a very nice relationship. And why is it interesting is that if we look at the spatial distribution of this gradient, right, or the spatial gradient of this phenomenon, we see that it's related or corresponding to a similar phenomenon which we know from the empirical data, where there is this distinct gradient in peak frequency. To summarize, we would like to put forward the structured flows on manifolds as the central object of interest for description of resting state. We show that how it arises from a connectome-based model and provide a link between the empirical data features and the mechanistic explanation. Lastly, what we find is interesting is that this approach gives us a way how to formalize descriptions which are already being used on a semi-formal level. That is, for example, those using the vocabulary of attractor landscapes and so on. So looking into the future, we would like to add more data features or describe our manifold with more data feature and relate to empirical data in terms of traveling waves. Also energy landscapes as extracted from the empirical data, and refined the frequency gradient when we are talking about EEG. Additionally, we would like to also include regional variability in our model as another source of symmetry breaking, where we would like to use the various cortical gradients from neuroanatomy or histology to inform the parameters of our model. And lastly, we would like to look into neuromodulation. We would like to refine the role of the subcortical structures and allow them to modulate the activity on the cortex and be an additional mechanism or source which shapes the manifold and the associated structured flows. So with this, I would like to thank you for your attention, and I hope